My name is Hyun Park from Chris in Daejeon. First of all, it's my great pleasure to give a talk to you in 60 Gender Summit Conference. My title of my presentation is Gender-Related Toxic Response to Chemicals and Nanomaterials. The outline is in the following. The first, I will talk about the reproductive toxicity by 2-bromopropane. And after that, I'll talk about the toxicity of nanosilver nanomaterials. And I'll summarize it. Nano research has been conducted by so many groups last 10 years. But the research results are very difficult, different and controversial. And so some researchers reported that nanomaterial has negative for health, but the other people report that the nanomaterial is effective for human health. And what makes these differences? And this, the problem is which nanomaterial is used for the in vivo test. All experiments are executed by standard materials, but physical chemical properties such as particle size and distribution and degree of agglomerations and so on and so on. And this picture is carbon nanotubes. The shape of carbon nanotube is typical different. And these induce different results from same experiences. So it's very important to use the standard nanomaterials. The purpose of nanosafety research is the determination of standard protocol for characterization and monitoring property change in physiological conditions and decrease experimental artifacts by the nanoparticle interference and detection of matrix effect and so on. Okay, as the first subject, I will talk about the male and female reproductive toxicity by 2-bromopropane. 2-bromopropane, known as isopropyl bromide, has crystal structure like this. This material used as cleaning solvents in electronic industry. 20 years ago, 25 workers had reproductive toxicity. And for male workers had aplastic anemia with aspermia or azoospermia. And for women workers had amenorrhea or ovary atrophy. And at then exposure duration time was 10 months. And to analyze this case, we started the research using the nanomaterials. This is testis toxicity of 2BP. As the dose concentration is increasing, and then the testis size is decreased. This picture A is the typical shape of testis, but the figure B is the sample after treatment of 2BP. And this black point is the germ, but in figure B, there is no germs. This is, no germ means there is dead point. And this is reproductive and hematopoietic toxicity of 2BP. As the dose concentration is increasing to 500 milligram per kilogram, the body weight decreased to 290s. And testis acoustic were observed to very high dose concentrations. The white blood cell also decreased, and the hemoglobin value also decreased. The value of hemoglobin induced the anemia. This slide shows the effect of 2BP on the female reproductive functions. As those concentration is increased to 900 milligram per kilogram, the body weight also is decreased, and the ovary apoptic also observed to at very high concentration. The S2 cycle also become longer compared with the contingent samples, and no pregnancy is observed in these at very high concentrations. This is a second subject about the gender differences in nanosilver toxicity. This toxic chamber for inhalation, this is number 1 and 2 and 3 is has nanosilver generator which can make the silver nanomaterials. And in this area is the rat in a wild cages. And this chamber is automatically controlled. 4 chambers is there, and high and mid and low concentration is possible. And this slide shows the change of weight during 90 days. The male animals shows the meaningful fact, but in female has nutrient effect. As those concentration very high, and then this weight is very high. But this middle concentration, in this case, is the weight is very low. This is lung function testers. Lung function tester is measured various parameters such as stridor volume, which is difference between inhalation and exhalations, and minute volume and frequency. This is lung function testers. And this slide shows the change of tidal volume according to the days. Male, this is male in the male sample is the tidal volume is not affect to the as the tidal volume is decreased, but in female case, the total volume is not effect on the its days. This slide shows the gender difference accumulation of silver nanoparticles in kidney. As the dose concentration is high, and then the accumulation rate of inhalation is also increasing, but the weight accumulation rate is 3 times larger than for man. In oral experiments also observe the same result. This means both inhalation toxicology and oral toxicology means have gender differences. This slide is silver accumulation in kidney. This is cortex in the outer medulla, in the inner medulla. This is female, this is male. The red color means accumulated silver particles. In cortex regions, the accumulated weight of female is similar as the male, but the out medulla there is no accumulation gold silver particles except in the boundary, and the inner modulus, and then the accumulation weight of female is larger than the males. And in case of male, there is no any silver accumulation. This is accumulation of silver in adrenal. This is a control sample. This is female, or this is male. In medullary region, the accumulation weight in female is larger than the males. In other areas, it's not found in silver particles. This is liver toxicity of silver nanomaterials. The abnormality of female is 2 times larger than males. And in female case, the necrosis and bile duct hyperplasia was found. This is the subject of my talk, the gender difference in clearance of silver nanoparticle from organs. This is experimental design for clearance. The used nanoparticle size is 10 nano and 25 nanometers. And the oral administration day is 28 days. And the concentration is 100 and 500 milligram per kilograms. The recovery time is 1 and 2 and 4 months. These slides show the remnant silver nanoparticle in the brains. In silver nanoparticle, in this case, silver nanoparticle is not cleared at all, and female is so similar. But in testes, also silver nanoparticle is not cleared at all. But in ovary, all silver nanoparticle is perfect cleared. This is summary. As a chemicals, 2BP is reproductive and hematophobia toxicity to both male and females. But silver nanoparticles accumulated preferentially female kidneys and adrenal 3 times more than male kidneys. Female is more susceptible to liver toxicity induced by silver nanoparticle exposure. And silver nanoparticles are biopersistent in brain and testes. And lastly, gender differences in the toxicity of silver nanoparticles exist. Thank you.